There are two ways in which an organism can reproduce. Many organisms, specifically bacteria, fungi, a few plants and animals, can actually undergo asexual reproduction. This is where a single parent produces an identical offspring from itself. Now, we saw this in binary fission at the beginning of the chapter. I think it was in the first video, where the cell basically divides through a process that's really similar to mitosis. And remember that mitosis was, the point of it was to produce two genetically identical daughter cells. So in asexual reproduction, the parent is identical to all of its offspring, and all of the offspring are identical to one another. The other process by which organisms can reproduce is sexual reproduction. So this is the one that you guys are probably most familiar with. This is where you get the fusion of two reproductive cells. Our reproductive cells are called gametes. Generally called sperm and eggs. And this process is called fertilization. So you get the fusing of those two gametes. Those cells then divide and grow into a multicellular organism. Some species, especially plants, can actually use both. There are some strange examples of animals that can do both or switch depending on the circumstances. We're generally going to talk about just plain old boring sexual reproduction, organisms that are just doing one and not the other. So I said that there were two different types of cell division. One was mitosis. We already talked about that. That is the process by which we produce most of the cells in our body, where we get genetically identical cells to the parents. So the other process is a process of meiosis. Now, meiosis overall is really similar to mitosis. And so when we get to the actual process, I'm not going to go into it in too much detail, but there's some very specific outcomes. And one of those outcomes is that mitosis reduces the number of chromosomes in a cell. So let's back up just a little bit. Humans have 46 chromosomes altogether. The 46 chromosomes are made up of two sets of 23. So we get one set of 23 chromosomes from mom, one set of 23 chromosomes from dad. And each of those chromosomes has a similar partner in the sperm and the egg. So we each get a copy of chromosome 1 from mom and from dad. We each get a copy of chromosome 2 from mom and from dad, and so on. The cells that are used in fertilization are what we call haploid. This means they have one copy of each type of chromosome. The cells that result from fertilization are what we call diploid. That means they have two copies of each chromosome, one from mom, one from dad. So during this process of fertilization, two haploid cells combine to produce an organism with the correct number of chromosomes. Now we need two copies of each type of chromosome in order to produce a fully functional, normal individual. So let's look at what this whole process looks like. So up here we have mom. She is going to donate an egg to this situation. We have dad, who's going to donate a sperm to this situation. As individuals and in all of their somatic cells, they have two copies of each type of chromosome. So if this was mom's set of chromosomes, she would have gotten one of each of these from her mom and her dad. Same thing with dad over here. He has a copy of each set of chromosomes, one from his mom, one from his dad. The process of meiosis occurs in the gonads. Those are the ovaries and testes, the reproductive organs of each individual, and they produce the sperm or the eggs. The sperm or the eggs are the gametes, which are our reproductive cells. So the process of meiosis reduces that diploid situation down to one. So now in each one of mom's eggs, she has only one copy of each type of chromosome. Dad has the same thing. His sperm has only one copy of each type of chromosome. Then, when the sperm and the egg fertilize, they produce something called a zygote. That's just the biological term for the fertilized egg. That brings back together those two haploid sets of chromosomes, and now the fertilized egg that's going to grow into the baby actually has one set of chromosomes from mom and one set of chromosomes from dad to be diploid again. So the process of meiosis is the process that's going to reduce that diploid situation of chromosomes down to haploid. Then the process of fertilization is going to restore that diploid. After that fertilized egg begins to divide, it goes through the process of mitosis, which is how we grow into a fully functional organism. So meiosis is only occurring in the cells that are producing sperm and egg. All other cell divisions in the body happen via mitosis. Now, this change from haploid to diploid is often very confusing for students. So if you're not sure what's going on here, get through the meiosis video, and then I'd like you to draw it out and actually see if you can follow these chromosomes through a few generations. Great. So the overall goal of meiosis is to produce our gametes, to produce our sperm and our eggs. Now, this allows offspring to resemble their parents, and they resemble both parents because they get some genetic information from each parent once the sperm and egg are fertilized. But no two offspring ever resemble their parents in exactly the same way. And this is because during the process of meiosis, mom's chromosomes get shuffled and dad's chromosomes get shuffled, and so you get new combinations in each of your sperm and your egg. So essentially, the egg that was fertilized to make you was kind of a mixture of your grandparents' chromosomes. Same thing with the sperm. The sperm that fertilized the egg that became you was sort of a mixture of chromosomes from your grandparents. And this is why we see families looking really similar. And you can often tell siblings, even if they're not identical, because they have similar features because they've come from the same combinations of gametes. So here we have John F. Kennedy. He was one of our presidents. And then we've got two of his brothers, Edward and Bobby. And if you look at them, you can see that they look similar. There are similar features between each of them that might allow you to recognize them as related. But because they have different combinations of chromosomes from their same parents, they look different. They look similar, and yet they look different. And that is one of the important outcomes of meiosis. There are two important outcomes of meiosis. One is that it reduces the genetic material in gametes by half. Now, if it didn't do that, if we started out with 46 chromosomes in each of the first two humans, and then they had a baby with a fertilized egg that contained 46 chromosomes and a fertilized sperm, an egg that contained 46 chromosomes and a sperm that contained 46, we would end up with an organism that had 92 chromosomes. And then in the next generation, we would have 184 chromosomes. And so if you don't reduce your chromosomes in your gametes, you end up doubling the genome every single generation. And that would just become, aside from not functioning, it would become too cumbersome. You could never continuously double your chromosomes and still maintain a functioning cell. That the cell just couldn't hold that genetic material. So we need that reduction because our gametes fuse every time. And so every time we produce gametes, we cut the number of chromosomes in half so that we get back our diploid situation. The other important outcome of meiosis is that each of the gametes differ from one another with respect to the combos of alleles that they carry. So remember that alleles are different versions of genes. Now, in that set of chromosomes you get from your mom, you might have gotten a brown-eyed allele and you might have gotten a blue-eyed allele from your dad. So then if you consider another trait, let's say you got a short gene from your mom and a tall gene from your dad, your offspring could end up with any combo of those alleles. So maybe they get grandma's brown-eyed and grandma's short stature, and maybe they get grandpa's blue eyes and grandma's short stature. So each gamete is going to differ just a little bit, but you're going to get certain combinations that stay together, which is why you get similarities, and you're going to get some differences, which is why siblings never look identical to one another. So let's continue talking about meiosis here. Remember we said that meiosis occurs in the gonads. Those are specialized cells in the ovaries and testes that actually produce the sperm or the egg. Now, keep in mind that we are talking about meiosis in humans, so we're always going to start with 46 chromosomes, 2 copies of each type of chromosome, so 23 types for 46 chromosomes total. Each organism varies in the number of chromosomes that they have. This is one of the things that distinguishes different species. So, for example, chimpanzees have 48 chromosomes. Some plants have hundreds of chromosomes. Hedgehogs have something crazy like 98 or something like that. So the whole process is dependent on the starting number of chromosomes, but the idea is the same. We're just going to talk about our 2 sets of 23. So there's a couple of terms that I introduced in one of the earlier videos that I want to review here. And this is the idea of homologous chromosomes and sister chromatids. So homologous chromosomes are chromosomes that have the same gene locations all the way down. They also have the same physical characteristics. So they're going to be the same length and they're going to have centromeres in the same place. Now they might not be 100% identical, and this is because mom's chromosome might have that brown-eyed gene, dad's chromosome might have a blue-eyed gene, but they still have the eye color gene at the same place. Maybe another gene is a gene for freckles. So mom might have a gene that says no freckles, dad might have a gene that says freckles, but they still have the freckle gene at the same location. Now, when those chromosomes undergo replication in preparation for mitosis, or in this case, meiosis, we are going to produce sister chromatids. Now, remember that sister chromatids are like photocopies of one another. They are 2 identical copies of the same chromosome attached at the centromere. Now, something that's a little bit strange here is when we count chromosomes, we actually count chromosomes by the number of centromeres. So here we have, on top, we have 1 maternal chromosome, 1 paternal chromosome. Down here, even though we have 2 pieces of DNA, we still only have 1 chromosome because we count the number of centromeres. So because they are connected, they're considered 1 chromosome. Now, I know there's going to be some confusion and someone's going to say, but there's 2 chromosomes. And I'm going to say, but there's not. There's 2 pieces of DNA counted as 1 chromosome. So right this minute, I need you to learn the difference between homologous chromosomes and sister chromatids. Remember, homologues have the same gene position, the same structure, but they can differ in their alleles. Sister chromatids, on the other hand, are going to be identical in every way. They're going to have all the same alleles all the way down, same physical characteristics. As part of this complement of 46 chromosomes, we have 2 different types. We have autosomes. These are most of the chromosomes in our cells. And then we have sex chromosomes. Now, each of us contain 2 sex chromosomes. These are the X and the Y. And these are the only ones where the homologues really sort of differ in physical characteristics. So we'll talk about those in the next video, but just keep in mind we have 2 sets of 23 to make 46 total chromosomes. That set of 23 is made up of 22 autosomes plus a sex chromosome, either an X or a Y. So this diagram explains the overall process of meiosis and shows us how the genome is actually reduced by half. So we will go through the specific steps of each of these in just a minute, but let's talk about what happens in preparation for meiosis. So just like mitosis, our cells are going to be in interphase. They are going to have each of their cells containing 2 copies of each type of chromosome, one that came from mom and one that came from dad. And in interphase, they are going to replicate so that we have sister chromatids, and then they're going to begin to condense. So this is all the same no matter if you are doing mitosis or meiosis. You're going to replicate your chromosomes, and then your chromosomes are going to condense. All the same processes are going on, the spindle fibers, the movement of chromosomes, etc. Meiosis, though, is a two-step process, and that's because we have to reduce the number of chromosomes. So in the first half, in meiosis 1, we are going to separate these homologous chromosomes. So we have separated the chromosome that came from mom from the chromosome that came from dad. In the second half of meiosis, meiosis 2, we are going to separate our sister chromatids. Now you can see now, compared to the parent cell, we've reduced our chromosome number by half. So in meiosis 1, we separate our homologues. In meiosis 2, we separate our sister chromatids and reduce our chromosome numbers by half. So where we had, just bear with me, 46 chromosomes in the parent cell, we now have 23 chromosomes in each of the daughter cells. You can kind of start to see where these chromosomes are different, or these cells are different, I'm sorry. Remember we said one of the goals was to produce genetically different gametes. You can already see here, this cell has green, this cell has yellow. So that's the first step in making different gamete cells. So we have gone from a diploid situation of 46 chromosomes to a haploid situation of 23 chromosomes. This is just a stepped picture of how this happens. So we have parent cell, we produce our sister chromatids, and then we condense. We separate our homologous pairs, and then we separate our sister chromatids. All right, so let's go through each of these stages in meiosis. Again, I'm not going to go through them in super detail because the molecular process that's happening is actually the same as mitosis. So instead, I'm going to point out the differences, some of the similarities and some of the differences. So interphase, we replicate our chromosomes. Then we enter the process of meiosis. So in your gonads, those cells are never going to undergo mitosis. They are always going to undergo meiosis. Now, remember we said that the cells that are produced by meiosis are genetically different. And in the last slide, in the overview of meiosis, I showed you one of those ways because we separate our sister chromatids. Another source of novel genetic information is a process called crossing over. And this is where our homologous chromosomes actually exchange some genetic information. Now, the reason for this exchange is actually so they're kind of holding hands. And they need to do this because they need to move together to the metaphase plate. And I'll talk about the mechanism of crossing over a little bit later, probably in another video. But the reason for crossing over is so that those chromosomes stay together. Now, the reason that they have to stay together is that we are going to line up along the metaphase plate and we're going to pull them apart. Remember, the reason for lining up is that basically to get your ducks in a row so that all the chromosomes are partitioned equally. So then when we get to anaphase, we are going to pull our homologous chromosomes apart. Now, I want you to notice something else. This is a third source of genetic variation. This is called random assortment. This is where your chromosomes that originally came from your grandparents, so in your mom and dad cells, line up in different combinations along that metaphase plate. So here, let's say the dark chromosomes came from mom originally and the light ones came from dad. So this cell now has a combo of that's going to end up to be mom, dad, mom, and dad, mom, dad. So there's another source of new genetic information. So right here in meiosis 1, we see two sources of genetic variation. One is this crossing over event. The other is this random assortment. Now, these could have just randomly lined up with mom, 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 and dad, dad, dad. We could have done mom, mom, dad, 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 mom. So depending on the number of chromosomes, and you can imagine if we have 46 chromosomes in there, you can have a huge number of possibilities of combinations in the gametes that are new mixtures of the parent chromosomes. So then we move on to meiosis 2. Now, this is another round of meiosis. At the end of meiosis 1, after telophase and cytokinesis, the cell enters a brief interphase, just sort of does a check to make sure everything's ready to go. The only difference here is that it does not replicate the chromosomes again. That would defeat the purpose of reducing the number. So brief interphase after telophase of meiosis 1, and then both of those daughter cells are going to enter another round of meiosis. Now, meiosis 2 is actually basically identical to mitosis. In this round of division, we are going to separate our sister chromatids, which, if you recall from mitosis, that was the point. So again, we line up along the metaphase plate, separate during anaphase, and end up with 2 daughter cells. Now, the ultimate result of meiosis is the production of 4 cells, each that are genetically different from each other. So if you take the time to look at these chromosomes, you will see all different combos of light and dark and crossing over. Okay, so here we have a light blue with a half light, half dark green. Here we have a dark blue with a half light, half dark green. That is representative of the different types of chromosome information that we have on each of those cells. So at the end of mitosis, I gave you a nice little summary of what happens. So I'm going to do the same for meiosis. So meiosis occurs as part of the process of sexual reproduction. It differs with respect to meiosis in that it takes 2 rounds of cell division, and that's because we want to reduce the number of chromosomes by half. Meiosis only occurs in our gamete-producing cells, the cells that produce sperm and eggs. Ooh, I see a typo there. Fixed it. All right, meiosis produces 4 daughter cells. Each of those daughter cells are genetically different from the parent and from each other. So even if you took 4 daughter cells from the same division, so say that was 4 sperm cells from the same division, and fertilized even the exact same egg, you would have genetically different individuals, just because the sperm's chromosomes are different. And then you have the egg's chromosomes that are also different. So we get a huge amount of genetic variation through this idea of meiosis and sexual reproduction. Now remember that in meiosis, the resulting cells are haploid, which means they have half the number of chromosomes, and then that diploid situation is restored during the process of fertilization.